Hi, everybody. My name is Anoushka Rajeshkumar. I'm a sophomore at Vista Del Lago High School, and today I'll be presenting my research on the application of different stem cell sources for cardiogenesis and osteogenesis. I decided to research this field because of an experience that I underwent a few years ago when I had to have a surgery that resulted in a pretty big scar. And the only treatment options that I was given for that scar were a silicone-based scar gel that did little to nothing or plastic surgery. So over the past year, I've been researching like scar treatments and wound treatments when I kind of stumbled over the general field of regenerative medicine, specifically stem cell therapy. And ever since, I've been working on this project with my mentor, Lana, and I'm really excited to share it with you. So without further ado, let's get started with the stem cell showdown. Let's start with some background. So regenerative medicine is an emerging field that focuses on the repair, regrowth, and regeneration of cells, tissues, and organs. Within that whole field, stem cell therapy is a sector that focuses specifically on stem cells for tissue repair and regeneration. So what are stem cells? Stem cells are unspecialized cells that have the ability to develop into different cell types, and they have two key factors that differentiate them from other cells. Number one, differentiation, or a stem cell's potential to specialize into a different type of cell, for example, a heart cell. And then number two is self-renewal. There are two types of stem cells, embryonic stem cells and adult stem cells. Embryonic stem cells are pluripotent, meaning that they have the ability to differentiate into a very wide variety of cells. And these were initially used in stem cell research, but they raise a lot of ethical concerns because the actual harvesting has to be done in utero and often kills the embryo that it's harvested from. That's why a lot of research has transitioned towards adult stem cell sources, which are more specialized, but they don't come with the same ethical concerns as embryonic stem cells. So today I'll be focusing on three types of adult stem cells: adipose-derived stem cells, bone marrow-derived stem cells, and induced pluripotent stem cells. And understanding the advantages and disadvantages of each type of stem cell is really critical for future applications in regenerative medicine. First, let's focus on adipose-derived stem cells. So ADSCs are derived from fat tissue, and they're typically obtained through a minimally invasive liposuction procedure. There is a large supply and availability of ADSCs, and they have the easiest harvesting process. Over here, I have a rendering of some ADSCs and then the liposuction procedure for harvesting. Now let's move on to bone marrow-derived stem cells, or BMSCs. So these are extracted from the bone marrow, usually through the hip bone via an aspiration procedure. These stem cells are usually less available, and it does have a more complex harvesting process that's harder on the patient. Over here, I have a rendering of a BMSC and then the aspiration procedure. Now let's move on to our final type, which I think is really cool. These are induced pluripotent stem cells, or iPSCs. These are actually created by reprogramming adult cells to a pluripotent state in the lab. So because they're pluripotent, they have a much wider range of application than the other two stem cell types because they can differentiate into a lot more types of stem cells. However, despite how cool these are, this is a relatively new procedure. So it's very costly and complex, and we don't know like the full side effects and different things that could happen with this type of stem cell. So now let's move on to the comparison of these sources for specifically cardiogenesis and osteogenesis. When we talk about cardiogenesis, we talk about cardiovascular diseases, or CVDs, and these are the leading global cause of death, and they're actually responsible for 32% of all global deaths. A lot of the current treatments for CVD focus more on managing symptoms through things like medication, surgery, and lifestyle changes, but they don't actually regenerate damaged heart tissue, so we need more innovation in terms of tissue regeneration. So let's get started with adipose-derived stem cells for cardiogenesis. My reference is at the bottom. It's a study by Dr. Leong, and the key findings of the study were that ADSCs can differentiate into key heart cells and reduce inflammation for cardiac repair. They've been shown to promote blood vessel growth and reduce apoptosis, which is just a fancy word for cell death. ADSCs also grow faster, age slower, and produce more cytokines, especially when compared to BMSCs. And cytokines are immune proteins, and the result of more cytokines is stronger immunosuppression, which is very necessary when considering like transplants or transfusions, because without immunosuppression, the immune system will attack these stem cells and it'll basically attack its own body. Now let's move on to bone marrow stem cells for cardiogenesis. So my reference is at the bottom, and this study focuses on an in vivo model on mice that found that BMSCs led to new heart muscle cells, which does indicate some regeneration. However, despite the initial repair benefits, the effects were not long lasting. And then two other clinical trials, Time and Poseidon, also showed no significant improvement in heart function after bone marrow cell treatments. Now let's move on to induced pluripotent stem cells for cardiogenesis. So my reference at the bottom found that iPSCs led to improved heart function by reducing inflammation, increasing anti-inflammatory macrophages, which are immune cells, and improving heart remodeling and regrowth. So iPSCs have shown greater improvement in heart functioning, specifically ejection fraction and fractional shortening than BMSCs. Ejection fraction is the percentage of blood that's pumped out by your heart, and then fractional shortening is the measurement of how well your left ventricle contracts during a cardiac cycle. Overall, for cardiogenesis, I determined adipose-derived stem cells to be the best option for heart regeneration due to its strong differentiation and faster growth, as well as its promotion of blood vessel growth, reduced inflammation, and secrete healing growth factors. While iPSCs do have a lot of potential in cardiogenesis, they do have a big tumor risk, especially for teratomas, because they are pluripotent cells. They can differentiate into more types of cells, which can lead to tumors. Additionally, the entire process for iPSCs is much more complex. Additionally, ADSCs grow faster than BMSCs, age slower, and offer more immunosuppression, which is safer for the heart to make sure that the immune system doesn't attack the heart. Now let's move on to osteogenesis, where we'll be considering bone disorders, which affect many million people globally. Just one example, osteoporosis, it affects 200 million people globally. In terms of bone regeneration, bones have very limited healing ability, especially in chronic cases like osteoporosis, and current treatments focus more on symptom management through medication, surgery, and lifestyle changes, but they don't actually promote long-term healing, which is why we need more innovation in this sector again. So let's start with adipose-derived stem cells for osteogenesis. My study at the bottom applied ADSCs in craniofacial defects and musculoskeletal disorders, such as osteoarthritis. And 10 studies with 307 patients indicate that ADSCs are safe, but it did show limited evidence for significant bone regeneration. The ADSCs fail to form bone in immunodeficient animal and human models, which shows that it's not as suited for osteogenesis. In terms of BMSCs for osteogenesis, my source at the bottom found superior bone forming ability as compared to ADSCs, which is likely due to its origin because BMSCs are harvested from the bone marrow. And then BMSCs are also capable of differentiating into osteoblasts and forming bone-like tissue at a higher rate than my other two sources. And then additionally, these have been used for cartilage repair and osteonecrosis, which is bone tissue death, and we've seen a lot of success. Finally, let's move on to induced pluripotent stem cells for osteogenesis. My study at the bottom found that iPSCs are capable of becoming osteoblasts and osteoclasts, which are the two types of cells that lay down the bone and then break it up. However, this was at a lower rate than BMSCs. And then it also found that iPSCs often lack supporting structures such as nervous system and vasculature, affecting its functionality and lifespan. And this is very crucial because without supporting structures, the cells won't actually be able to survive. And then regeneration is complex and can also be disrupted by local and systemic factors specific to iPSCs. Like I mentioned earlier, iPSCs have a large risk for tumorigenesis because they are pluripotent. Overall, for osteogenesis, I determined BMSCs or bone marrow stem cells to be the best source for bone regeneration due to its superiority at differentiating into osteoblasts and osteoclasts. Additionally, it's effective in bone defects, cartilage repair, and osteonecrosis or bone death, with a strong track record in promoting bone formation. BMSCs also consistently produce bone-like tissue and integrate well in healing, unlike ADSCs. And iPSCs, again, they have a lot of potential in osteogenesis, and we have seen positive studies, but they just have such a big tumor risk and also functional limitations due to lack of vasculature and other supporting tissues. So with that, I can conclude that ADSCs are better suited for cardiogenesis due to its strong differentiation potential, faster growth, and immunosuppression, while BMSCs show superiority for osteogenesis because of its good osteoblast and osteoclast differentiation. There are a lot of limitations for all three types of stem cells. Like I said, ADSCs and BMSCs show promise, especially in their respective categories, but efficacy does vary, like especially study to study. And iPSCs, so they are very versatile and they're so cool. They pose a lot of risks like tumorigenesis and production complexity. So overall, more research is needed to optimize these stem cells and enhance tissue regeneration strategies. Thank you so much for listening to my presentation. Feel free to ask any questions.
Thank you so much, Anoushka. What an interesting study. I had no idea that there were so many different types of stem cells. I had a particular question about the induced pluripotent stem cells. I'm curious if you could walk us through how exactly do they induce the cell to differentiate in a certain way? Is it by like injecting chemicals or like how does that work?
Yeah, so in terms of induced pluripotent stem cells, to the slide. In terms of induced pluripotent stem cells, they're actually harvested like as normal somatic cells from the adult body, and then in the lab, they're differentiated. I don't have the details on the exact steps of differentiation or sorry, of pluripotency, but it is still like a relatively new procedure, so there haven't been as many studies on it, but I think in the future it's definitely going to continue to grow. And since it is such a big thing in stem cell therapy, I think a lot more research is going to go into it, and then we'll find out a lot more.
That's cool. And we have a question from Sarish. What was the hardest part about conducting this research?
Yeah, so I think the hardest part was since I am a high school student, like a lot of the articles and journals that I found specifically for stem cells, especially like at this level, were very complex. So I really took me like a lot of time to go through and actually research everything within the articles. So honestly, it was just a huge time commitment, but I did really enjoy it because I loved learning about it. But I think just understanding it and then being able to simplify it and then like present it was that was a challenge for me.
And do you think, sort of similar to the question that I asked Sarish earlier, do you think this is what you might want to like major in or what you might want to go into in the
Yeah,
future? Or, yeah, what are
I
your thoughts?
mean, I currently my goal is to become a doctor. I'm not entirely sure on what specialty. I was considering oncology, so this obviously is very related to oncology. In college, I want to go into something in the medical field, whether that's just like general biology or molecular biology. Yeah, but this is definitely something that I want to pursue.
Super cool. Thank you so much. This was a great presentation, Anushka.